So this work is about mainly a nanoscale, like microscopy images, colorization using neural networks. So mostly in biology and science, it's very important to have like microscope devices. Like microscopes are there, like very important. They help us to see a nanoscale or microscale objects under the microscope, like for us. But most microscopes, like they make images through the electrons, like rather than using a kind of a visible light from the photon. So the images, like the form is like grayscale or like single layer images. So these single layer images contain much less information than the same version of a colorful image. So using this single layer image or grayscale images out of the box might create like a gap between the experimenter or like the researcher and the audience. So, basically the colorization task is a very common task among the researchers, but it's very time consuming and expensive task. So the common methods are like using softwares like Adobe or ImageJ or Fiji, especially in the biology part. Even though these softwares came with some semi-automatic methods, they basically need like a massive user input and still like time consuming. Like I remember there was an image took around a month to color, like to give a color, because sometimes even like artists just might have to draw them. And the other thing is like most these images, they don't have one color. Like, so I might give a color like as a black or as a green, as a red, or as everything. But we know that these images are a direct or like indirect representation of real world images. So it might come out with bread or like dumpling or coffee or some toys. So how do we find the relationship between these real world images and these SEM images, and we give them color? So we basically come up with two solutions. So the first one is like end-to-end CNN model. So what we did here is like we contributed like the first data set of SEM images. Then as you know, like deep learning is a quite zoo of architectures. Like we've got a lot of architectures. Then when we do like a kind of prediction, you basically have encoder, then you have like a fusion, a decoder, and this like it goes like this. So we trained like this model on our own data set. We collected like from our lab and from like internet. Then we train the model, the encoder is just trained on our data set. Then we've got a pre-trained model trained on ImageNet, so as we use it as a feature extractor. So you can find a relationship between the microscopic world images and the real world images. So like what does it have? So what is the content and like how does we give it the color? So the basic structure is like we have an encoder here, so the encoder just takes an input image and kind of extract the middle level features like colors. So we have inception module here, it's like a feature extractor, so to obtain shapes and to find relationship. So we have a fusion layer like to merge the encoder or like concatenate the encoder with our inception. Then finally we have like a decoder give us output. So basically we use LAB color space here. So we've got like, we give it as L channel, a lightness channel, as input, then the model predicts AB channel. So finally, like we just again sum them up, then we can get like RGB image. So even though we don't have a ground truth for this, so it's very hard to find like what the loss function looks like. So because you don't know the ground truth of like the image, you just might train it on one data set, but still you kind of get lost in the middle of nowhere because like they don't have one ground truth. So we just like predict like one image, like one color in each pixel, and we try to find like its ground truth as like from the previous, like from the given image, like from the data set that we have it. So these are like outputs, like an output for one like just image. So the first one is like it's bacillus, then we have a cilia. It kind of have like a yellowish color. Then we have it a wood surface on the said like column. Then we have a nuclear membrane, stamens, and we have it like a leaf surface. So like a leaf surface is just obviously, I don't know, it's like it should be green, but I don't know what it looks like there. Like just it's green. Then the leaf surface has like a green color. It should have it like because it's obvious a leaf is a leaf, right? So sometimes it's very computationally it's expensive to give a prediction task for a model. Like you give it one image, then the model kind of predict like a lot of colors. Then even though it give you a color, but the color doesn't make sense because let's say you have red blood cell, but the red blood cell might have like some blue color, which is it's not true. Or a plant tissue or a plant surface might have some yellowish or like some less gray color, which just doesn't make sense. So we try to modify like our end-to-end standard model rather than training it like on whole data set. We try to redesign it to accept grayscale image and a colorful reward image, and like as kind of a style transfer model, but like not traditional style transfer model, because just the microscopy images only get the colors from the reference image instead of getting like shape or something else. So what we do here is, let's say as reference you give it a bread, then you have it like you train it on the train, then you during inference time we save weights, then we transfer this weights for the final for the your style image, which is the content image. So these are like just some results from our data set, or from our experiment. So you have an input, then you give it for the first image we have it as a bread, then the images get colors from image, the shape from the first image. The second one is as a kind of a plant tissue, then we have it we give it a reference as neurons, then they can get a color without changing anything. The third one is red blood cells, so you just give it red tablets, then the color is just transferring into that. So the other obstacle was sometimes you might have a kind of substrates when you do experiments. So using some models or even softwares, they just get mixed the substrates with the main objects and the image. So what we did is you just give without doing anything on the model. So the model will be try to understand what is in the reference image. Then you kind of get the final output as color, different colors. The substrate will have another color, and the main objects will be another color. So it's just here, you can see the chromosome on the left side, then the DNA helix on the right side. So this is another result. So we wanted to do a kind of analysis for it. We're pretty much curious, how much our model can be smart and how can it fool people. So we took 10 participants from lab. Then we asked them which one is from our model and which one is from colorized by human beings. So our model was try to it was pretty much smart to cheat people that the average people thought the image were colorized by humans, while the models were from our data set, and vice versa from our model output. So the previous methods comparison and this is our image, our final output. Okay, so yeah, sometimes the images you don't have to just, because they have a 3D structure, so you just give them a color, then you might miss some contents. Then what we do here is just try to zoom in up and zoom up, then see what's going on over there. Yeah, thank you.
I think that was a good presentation. From an applications perspective, what would you see the colorization, what would it be useful for?
Oh, you mean why do we need them, right? The colorful images. It's it's very important to have colorful images, because sometimes you do experiment, then the real world image, which means the one you did as experiment, is something that you wanted to see. But when you have a grayscale image, you don't see the objects, you might mix them up, the substrate with the main object, or even 2-3 objects in one image might be combined and mix it up. So you have to just see them as you have to give them a different color to represent this.
So since your data has different layers, what do you use for labeling them, and what guide do you use for labeling them? Because when you have medical data, you have problems with labeling. And how do you get the good quality for labels?
Labeling?
Yeah.
How do we label the data?
Yeah.
Oh, we don't label the data. What we do is you get a value from a pixel. So you just extract that pixel in color space. You just get L and AB kind of space. So you just don't label them. Saying you kind of how do you say, like it's like make like a group of like labels. It's like, I mean, values. So you say like same grade values will have the same AB values. So you just find it this way. You don't label them.